start recording as we had permission to record, and then I'm going to give an introduction for today's talk. I think this is the second talk of CGDG for this year, and I would like to welcome Yvette Steyn, my colleague from my days at the University of Georgia. And the presentation title today is Genomic Evaluations for Single Breed, Multi-Breed, Crossbreed, and Crossbreed Predictions. So just a few words about Yvette. Yvette is South African. She's currently a postdoc at the University of Georgia. She completed her master's in 2012 at the University of Pretoria. She was working on selection criteria for feed efficiency in beef cattle. After that, she worked for 5 years for SA Studbook, where she was part of the team responsible for genetic evaluations of most of the breeds in South Africa, which included over 20 beef breeds, 12 small stock breeds, and 3 dairy breeds. And then finally, she recently completed PhD with Ignacy Mishtal in 2021. And she will today talk about her project. So Yvette, whenever you want, please, the floor is yours.
Thank you, Ivan. So as you said, a lot of my presentation today was part of my defense last year, but I have adjusted it. So first, genomic prediction has become popular worldwide quite quickly. And as we know, the reference population is one of the biggest elements that determine accuracy of the evaluations. There are specific requirements that it should meet, and the big one is that it should be a big reference population. But in the meantime, we've determined that just big is not important enough. It has to have other elements in it as well, including having phenotypes. It doesn't help if you have 1 million genotypes and absolutely no phenotypes. The relationships matter. The reference population should be related to the validation population. Within the reference population, it's ideal to have them less related to each other. It should not be biased in which ones get measured. You should have it from a whole spectrum of the worst to the best. And it should be updated constantly. So we have seen that as the generational distance increases from the reference to validation population, the accuracy decreases. So you can't just use an old reference population forever. It's also ideal if all herds are included. This counts for any kind of evaluation, multi-breed or normal with inbreed. So with inbreed is how we usually know it. So we would have a reference population that has animals from one breed, and that information gets used to predict genomic predictions for animals within the same breed. Multi-breed is a bit different. So here you will have a combination of breeds in the reference population, and you'll use that to make predictions for animals that are within this population. So it's not a different breed, it's one of the breeds included in the reference. So from previous studies, there's been little to no benefit, which has been a little disappointing. And it's been inconsistent. Sometimes Bayesian methods will work better, sometimes GBLUB. Sometimes different breeds will perform better and different traits. It's been hard to predict exactly what will react differently. The SNP density is important, and studies have shown that once you go over about 50K, even if you go to whole genome sequencing, there's not much benefit. It can even decrease the accuracy. Many of the Australian breeds have worked on SNP prioritization, that they use fewer SNPs and they obtain similar accuracies. Some of the breeds may be much bigger than the smaller ones. Generally, the changes observed would be in the smaller breed. Sometimes it's positive. When the small breed is way too small, it really gets warped by the larger breeds. A crossbreed is different, where you use a breed, one breed in a reference population, and you predict animals from a completely different breed. This can be similar to a multi-breed scenario where you have multiple breeds in the reference population, but you use this information to predict animals that are not included in the reference population. I'm making this definition clear because I have seen from multiple studies that they use these terms inconsistently. Sometimes they will talk about a multi-breed, where according to this definition of mine, it is in fact in a crossbreed. So be careful when you read conclusions, just make sure of the definition first. The justification for doing this is to increase the reference population for multi-breed scenarios. And lack of funds. Sometimes one of the breeds will have less funds and resources, specifically in some countries. A lack of phenotypes that can be due to bad recording, or just that you want to do a completely new phenotype that another population has been doing for a long time. It has allowed international collaboration. From South Africa, we've tried this a few times. So the Wagoos has joined the Australians, and some of the sheep breeds, we also looked at possibilities to do that with Australia. This has unfortunately been really disappointingly unsuccessful. And reasons for this is discussed widely by Yvonne Vinkis. She's done extensive research on the reasons, both theory and practical. And she was part of the group with Andres Legara that did a really nice review paper on this that I suggest. The linkage disequilibrium does not persist across breeds. As time went by, these segments have broken differently, and it won't be the same. And even if LD is the same in the different breeds and we can capture the QTL differences may differ. So this we can see clearly with DGAT, where it's different in Holsteins and Jersey. Even if QTL effects are exactly the same, the allele frequencies may differ. The number of QTL is important. If there are fewer QTL with larger effects, it tends to be a little bit more successful. G by E will influence how it's expressed, and non-additive factors such as epistasis and dominance. Crossbreed is a different kettle of fish, and this is where we use one breed and combine it with another one to produce something new. This is the simplest crossbreed you can get, but it can become more complicated, as we know, like rotational crosses and composite breeds over time. Crossbreeding is essential for the industry in pigs and chickens. It's a crucial part of their breeding strategy and has been for a long time. Here in our group at UGA, Natalia Lici is passionate about pigs and becoming better and better. I'm sure you're going to hear a lot from her in future. And Jennifer Richter is the up-and-coming chicken person in our group. The sheep and beef have mostly been using crossbreeding for breed complementarity as well as heterosis. That's common in their system. The dairies, New Zealand has adopted that approach long ago, but it's becoming more common in other countries, including here in the US. Reasons include to increase some of the diversity that they have lost and fertility and health that has come along with it. Also more advertising from other European breeds that have become more appealing here. A newer one is beef on dairy. On this one specifically, the Irish group has done extensive work on, and they even have selection indexes to choose the bull that's more appropriate for your dairy herd. The benefits of crossbreeding and multi-breeding is to add genetic variation. This has become more important, as I said, with especially dairy or small breeds. This improves fitness. You can use it to transform your herd, which I will explain a little bit more later. And by including crossbreeding animals into your reference population, you can increase the accuracy of the component breeds that it is made of. And similarly, if you add crossbreeds to a reference population, it can increase the accuracy of crossbreed animals, especially if your focus is on crossbreed performance, such as pigs. Methods that have been tried include a single matrix, which is the simplest one, and most compare it to this. You just add everything together into a single matrix. Multiple traits has been better than just combining it all. So Linky Magedlela did this on the Finnish dairy, and others were done on other dairy cattle here. Breed-wise allele frequencies would take into account that different breeds are different, and it would adjust the G matrix accordingly. She also did it on the Finnish, and Daniela Larenko did it on pig populations here. And Nasir Mogadar has this recent study, and they had an interesting approach to do this. And while for the others it didn't matter so much, it did make a difference in their study in sheep. Accounting for LD can either keep the accuracies more or less the same and potentially increase it. When an increase was found, it wasn't more than what you would achieve if you just did multiple traits. Multiple G matrix is something the Australians like to do, where they have one G matrix that contains the SNPs that are less important and a separate one that has prioritized SNPs. Breed origin alleles, mostly from the Dutch and Danish people. And this is particularly important in crossbreeds and has been good for them. the improvements when found was usually when the breeds were very different. And breed proportions was one of those that I approached, and Paul van Reyden did it here in the dairy cattle. And it was also done in simulated rotational cross by the Danish. My first study was on genomic predictions in purebreds when I use a multi-breed genomic relationship matrix. The objective of the study was to evaluate the accuracy of multi-breed genomic prediction when using the same SNP effects for all breeds, and when obtaining breed-specific SNP effects by treating the markers as non-shared. I had a simulation study with 5 breeds, a decent heritability of 0.3, 29 chromosomes, 1,000 QTL. I've also done one with 200 QTL, and my conclusions were not different. I had 45,000 SNPs and 4 replicants. I used BLUP-F90 software, which is basically what I use for everything. And I also had a 9,000 SNP scenario where I just picked every 5th SNP. I didn't have all animals genotyped, so I did a single-step GBLUP, and I randomly selected animals from the earlier generations and included the whole last generation. This is a visual representation of what I did. The more important one ending up that each breed has about 15,000 animals, of which about 7,000 were genotyped. This is a PC plot of one of my replicates, and it shows that the breeds are different. Breed A and E are a little bit more similar, but it does differ in one pane. When I used SNP effects from breed A, I could predict A well, but not any of the others. The other accuracies were really very poor. If I just did a traditional BLUP, it was much better than what I achieved here. And this pattern was the same of any scenario, regardless of breed. Even when I added 2 breeds, 3 breeds, or 4 breeds together, I would increase the accuracy of the other one a little bit, but not much. I had the shared scenario. So this would assume that the SNP effects are the same in the populations. And on this left side is my SNP file. And that looks the way we always have it. So all the breeds would have overlapping SNPs, and this gives me a G matrix in which all animals are related to each other, regardless of what breed. It's also centered around allele frequencies based on the whole population. Then I had a different one where my SNP file was much bigger. So the first breed would have SNPs from 1 to 45,000, and the next one would have missing SNPs up to that point, and then start, have 45,000 SNPs, and then missing, as you can see. So this way I would get a G matrix in which animals within a breed are all related, but those across breeds are not. This automatically scales it according to allele frequencies within breed, as the program ignores missing SNPs. When I used this method, regardless of single breed, non-shared or shared, when I had 45,000 SNPs, it didn't matter what I did. The accuracies were all the same. But if I decreased it to 9,000, then I started to see an average drop of 0.08, going from single breed to shared, while my non-shared scenario stayed stable. I even did this with 1,000 SNPs, and then the difference was dramatic between single and shared, but it still remained stable for the non-shared scenario. The question arises of how to treat crossbreeds, because in this scenario, you have to classify a breed into an animal into specifically one group. You can't have crossbreeds. So a suggestion is to take crossbred animals, determine which alleles come from which breed, and then overlap it with the relevant one. So this crossbred animal 1, the first SNP came from breed X and the other 2 from breed Y. And you can do the rest similarly with how many breeds you want in there. You can also reduce the SNPs that you have. So instead of having a long SNP file, you can determine which SNPs are important in all of the breeds and overlap them. And then have breed-specific SNPs that are non-overlapping, that accounts for differences in different breeds while accounting for the similarities of them. In conclusion, the SNP density was important in my study. And mine allowed you to do one single evaluation, yet treat the breeds separate as if you did them separately. It keeps the accuracy stable, and it is computationally viable. Although my SNP file is huge, the G matrix is the same size, and the non-shared scenario has many zeros, so it can even take faster to get results with my method. I still advise single. Wherever you can use single breed, rather do that instead of multi-breeds. The next study was on indirect predictions for milk yield in crossbred Holstein and Jersey cattle here in the US. Here is an example of what I talked about with herd transformation. In this situation, I am a Jersey herd and I want to become Holstein. So instead of selling all of my Jerseys and buying new Holsteins, I can make use of heterosis along the way and do it in a cheaper way. So I would take my Jerseys and mate it to a Holstein bull and get an F1 that's 50-50. I then take the F1 and I mate it again to a Holstein bull, and now I get a 75-25. And so I continue this process until eventually I get an animal that can be considered as pure enough. Different associations have different rules, how many generations they allow, if any. And this still brings in some variation from outside without changing the phenotype within the population so much. They look similar. The objective was to evaluate the predictive ability and inflation of indirect genomic breeding values for crossbred animals when I use SNP marker effects from different reference groups or using the breed proportions approach. I had about 7 million animals with milk yield and 20 million animals in the pedigree, 45,000 markers. For the Holsteins, I had about 300,000 genotypes, but for computational reasons, that was just too many. So I took the number of animals that had both genotypes and phenotypes, which ended up being about 90,000. My Jerseys, all of them 41,000, and the crossbreds were only about 22. If you look at my data though, you look at the numbers and it's a lot of genotypes, but then you run into this problem where you see that most of these animals don't have phenotypes. This is a bit of a concern, and it's a problem for all of those, not just for my data set. My Holsteins, all of them had phenotypes, but my Jerseys, only about 10,000 of them had, and the crossbreeds, less than 2,000 of them had. My validation population was selected as animals that had only one lactation and was born within the last few years of data. I say within the last few years because I selected it in such a way that I had about 20% of the animals that had phenotypes. I think that is a reasonable validation population. When I look at my crossbred animals, in terms of their proportions, they leaned a little bit more to the Holstein side. They were 59% Holstein. However, my validation was a bit different. It leaned a bit more towards Jersey. It was almost 50-50. In my valuation, I used single step, and I had many genotypes, so I had to use APY. I back calculated to get the SNP effects from the GEBBs, and then I used indirect predictions that I call DGVs from these SNP effects. My predictive ability was my measure of accuracy, which is the correlation between the adjusted phenotype and the DGV. I had different reference populations for SNP effects, and the first one was using all of my Jersey animals with a random core of 15,000 for APY. Then I had my validation populations, Holsteins, Jerseys, and cross separately. This next scenario used all my Holsteins with a random core of 22,000, or only the crossbred animals, which was few enough to not need APY. Then I had a scenario where I combined all my Jerseys and all of my Holsteins with a random core of 25,000, and a mixed scenario. And in this one, I wanted all of the groups to be represented equally. So I used all of my crossbreds, then randomly selected about 22,000 Holsteins and about 22,000 Jerseys. My last scenario was breed proportions. This is more comparable to what Paul van Reyden did. So every animal, I had their breed proportions. I would know the first animal over here at 80% Holstein. So I would get the DGV based on Holstein SNP effects and multiply that by 0.8. The Jersey, the DGV based on Jersey multiplied by 0.2, and then I just add those together. So this is a kind of crude measure to do it, while still accommodating for the fact that they're not all 50-50. When I look at my predictive ability, and I used only crossbred animals, I got an accuracy of 0.5 for the crossbreeds, which is not bad. And the purebreds could be predicted by about half as much as the crossbreeds, which makes sense. When I looked at the purebred animals, when I used Jersey to predict Holstein, or Holstein to predict Jersey, the accuracies were quite low. Much lower, or considerably lower than if I used crossbreeds to do this. With inbreed, about 0.48 for Jersey and 0.45 for Holstein, the Jersey could predict my crossbreeds a bit better, possibly because it was leaning more towards Jersey. And the Holstein could predict it by 0.47. When I combined the breeds, I got a bit of drop in the Jersey breed and not change in the Holstein, which corresponds to previous studies where changes would be observed in the smaller breed. And this also predicted the crossbreeds as well as if I just used crosses. When I used equal proportions, I didn't get as good accuracies for the crossbreeds, and the accuracy for Jersey dropped a little less than Holsteins, probably because the reduction of genotypes from the Holstein to the mix scenario was much greater than the Jerseys. Breed proportions was my last scenario, and that was the least successful of them all. This contradicts Paul van Reyden's study, where he found similar accuracies or slightly higher, although less bias if he used breed proportions. When he broke his crossbreds up into different breed proportions, he got lower, the lowest accuracies in the breed proportion group that was about 50% to 75%. My validation is about 52%. So this does not necessarily contradict his findings. This is looking at the inflation. So this makes the assumption that everyone's intercept was at 0. And this will give us an indication of inflation or deflation. The mixed scenario is perfect. There's no inflation, no deflation. It has a coefficient of 1. When I used the crossbred animals, it was a little bit deflated and a little bit inflated when I used breed proportions, but it was inflated much more as I used other scenarios, with the Holstein being the most inflated. Conclusions from this studies, I find that breed proportions is not necessary to obtain accuracies. My mixed scenarios had less accuracy, but didn't have inflation. My breeding objectives are important. So what I mean here is, if you want to become Holsteins, it makes sense to use Holstein SNPs. If you want to become Jersey, use Jersey SNPs. If you are just planning to compare the crossbreeds to each other, it doesn't matter as much what you do. If you want to bring it back into the population, it could be a different scenario. I had small validation populations for my crossbreeds, about 350. So results may differ once I have more data to do this again. Now we've looked at crossbreds and multi-breed, but this is also within breeds. The US population is huge and is always treated as within breed. This study looked at the effect of population stratification, the genome changes, and genetic redundancy on the diversity of the US Holstein. When we see the Holstein population, we know that there's been a handful of bulls that have been used extensively all over the world. And this has increased inbreeding, and it has produced a concern about genetic diversity. If the animals are not diverse enough, it will struggle to adapt to both environmental changes and consumer demands. We look at the Holsteins as one more homogeneous population. But perhaps if we look into it closely, we could separate it into different populations that are genetically a bit different from each other. And if this is the case, it brings a question, how did they become different from an older population? There are different ways in which you can change. Allele frequencies change over time. And this first one is the way we always picture it, selective sweeps. This happens more if there are few genes that have a large effect. If it's favorable, it can become fixed quickly. It will increase in allele frequencies, and the others that are important will then soon follow. But there's another way, that's polygenic shifts. This is more the case when we work with the kind of traits that we use, which are highly polygenic. In this case, there's not a strong selection on any particular SNP. In other words, none of them are expected to change much, although the phenotype will change overall. Selection is a big reason why this changes, but there's also hitchhiking. So this would be an allele or gene that has increased in frequency, not because it is important, but because it is close to another one. This has increased genomic inbreeding because it leads to greater sections of the genome that are more similar. Random drift is something we sometimes forget about, but it can have a big effect. And that brings me to genetic redundancy. What this is that there's basically many genotypic pathways to achieve exactly the same phenotype. There are many variants that are all beneficial, and a different combination of them can come to the same breeding value. So there's more than enough beneficial variants. This is because some traits are highly polygenic. In this situation, you can see I have 3 hypothetical animals, all with exactly the same EBVs, But if you look at their genes, it's a different combination of them, different magnitudes, and they're all added together. With this, to make it a relatable example for us, let's say that we are all going to participate in a virtual challenge. So within a month, we want to all complete 50 kilometers. And you can do it at your own time. You can do it in different intervals. You can use cycling, walking, whatever you want. When we come together at the end of the month, we will see that all of us have achieved that goal, if we have achieved that goal, in different ways. And that's the way this works. And once you really see it, it becomes obvious. Another reason is that some genes will have the same function. Some genes that have a bigger effect, one might be there and it gets expressed, but when it is not present or it is recessive, then another gene will simply come and take its place. This has been seen in more studies where they try to silence specific genes, only to find that nothing has changed. Another big one is epistasis and pleiotropy. Some genes will have an effect on many different ones, and one gene will have an effect on another one. This makes it kind of fun and interesting because now it's not just an additive effect, it's different kind of scenarios. So my objective here was to investigate the stratification that occurs within the US Holstein and the role that genetic redundancy contributes to these differences. My data was up to 2014, I only had type traits, and I had to pick animals that I considered to be selection candidates at that time. So I chose sires of animals that were born after 2010 with at least 25 progeny. My idea here was that these are probably the animals that I would see on a catalog. The females, I got those that were measured in 2013 and 2014. So they have been around and are more likely to be around again or be selected from. That left me with about 20,000 selected candidates. And then I used k-means clustering on the G matrix to divide these animals up into 5 clusters. When you look at the PCA plot, you can see that they are different, they are separated. However, as we would expect, there's quite a bit of overlapping here. Some animals that are hard clustered that are a bit more fuzzy in reality. A way to see whether my clusters are truly different is the reduction in the sum of squares that's obtained through k-means. So once this trend starts to decrease in how fast it increases, that's about how many clusters are appropriate. So here from about 5 to 7 is still appropriate, and beyond that, it's unnecessary. Then I also looked at the inbreeding. So I used hypothetical matings with pedigree inbreeding, and I determined what would be the theoretical inbreeding I would get from these matings. In cluster 4, it didn't matter what I did, the inbreeding was quite low. But for the others, the inbreeding within cluster was larger than average inbreeding across cluster. This 4th one, the big reason is that it is the larger population and it contains animals that are diverse enough within that group to still obtain lower inbreeding. I also used the fixation index as a measure. So this would use the allele frequencies across the clusters in this generation. This is the distribution of the FST, which as we expect, most of them are small and then some of them are much bigger. On average, it was 0.03. And this is about half of what they found with French breeds, comparing different breeds to each other. This is a distribution, and although none of these SNPs actually reached significance, either with the adjusted Bonferroni test or with the FDR, those 2 at the top came quite close. Another way I measured it was the additive genetic correlations between groups. And for this, I used a new method that was proposed, and it essentially looks at the expression of a genotype in genetic backgrounds of different populations. So this is how it works. This is the point of comparison. So I'd use the females of one of my clusters to get SNP effects, and then I use the males as my validation population to get DGVs. Then I would use the females of another cluster, get SNP effects there, and use it to predict a DGV for this previous cluster. The correlation between these populations is then the correlation between what I got with this first scenario and with the other one. And I keep doing that with the other clusters as well. Because of this, of course, I will get 2 different correlations for each relationship, because one would be the expression of population 1 in the background of population 2, and another one would be the expression of population 2 in population 1. Also, these correlations would be underestimated because the accuracy of it is not taken into account. So I used Kalo's method to adjust it. And if I consider the heritability as the same across all the groups and the reliability as the heritability, then my adjustment factor is just 1 over the heritability. Here you'll see the genetic correlations. So what I can point out here is that on average, the correlations between populations was 0.75. But there are some high ones. Cluster 1 is quite highly connected to cluster 3 and cluster 4. But overall, cluster 2 is least related to the rest of them. Now that I've determined that there are some differences, at least, I wanted to see how these clusters have changed over time. How have they come to be the way that they are? So to do this, I defined this animals that I selected as generation 10. Then I got the parents of that generation and I made them generation 9. And I took the parents of those, generation 8, up until generation 0. So what's important to note here is that there are overlapping generations. An animal that's a parent in generation 9 may also be a parent in generation 8. On top of that, as we trace the families back, they will be overlapping between families because many animals are related. When we do that, we can see from generation 10, we have about 3,000 animals for them all. And as I trace it back to generation 0, it becomes smaller and smaller. The smallest group is 135 animals, and this is enough to still look at allele frequencies. It would be ideal to be bigger, but it's decent for this purpose. If we look at this generation 10, there are no animals that occur in more than one family. But as we go back, there are animals that do overlap among families. So when we get to generation 0, about 83% of the animals occur in more than one family in that generation. And this is also shown here in the Venn diagram, where 108 of these animals in the past are overlapping. They're shared among all of these families, and only a few are specific to each family. This helps to give us a more homogeneous base to compare to, because we know that they were all kind of similar from the beginning. So why are they different now? The allele frequencies, this is the absolute change from generation 0 to generation 10. As expected, most are not changing much, and some do change more. I wanted to visually observe these markers to see where there has been change differently in different families. So I got selected genes. Some of them were based on a study by Lee Ma, who looked at those that have changed the most in the population. Then I also wanted to see which ones have changed differently across my families. So I looked at the variance of change across them and the range. So the range would be the family with the least change and the family with the most change, that difference. In the plots that I'm going to show you now, the blue marker is the selected one that I'm looking at. And the red ones are the 20 ones that are closest to it. So on the SNP chip, the other ones that are before it and after it. Because I have 45,000 SNPs, this distance might not be very close, but it's still closest to what I have. These were 2 of the genes that were determined to change the most. And you can see here with the AVEN gene, all of them change similarly, which is expected because it's changed in everyone overall. When we look at USP though, family 1 and family 5 starts to change direction. So this could be due to epistasis, where another gene has become important somewhere else and it is connected and it turns. I find the behavior of the surrounding SNPs interesting because they are forming 3 different clusters there. I looked at DGAT, not because it changed the most, in fact, it didn't change much at all, but because it is important. And when I look at this, I can see that family 2 is quite different. There's a sharp decrease in that family specifically. So there's an antagonistic relationship between milk yield and fat. And at some stage, there was a bit more of a shift to change the fat content in dairy. You can also see with the changes around it that there's some hitchhiking where they follow the same pattern. When I looked at the variance, I could see that these family 1 and 5 also changed direction quite sharply. And family 2 and 3 are similar. And family 4 didn't change so much. What's interesting here also with hitchhiking is hitchhiking doesn't have to mean that they both go in the same direction at the same rate. They can go in opposite direction if they are in different phases. The range You can see in family 1, there's a clear difference, and this looks like a selective sweep. It's important to remember that the SNP I'm looking at might not be the one of interest, it might be the hitchhiker, and the others are different. Now I have cherry picked, so I've specifically looked at ones that are different, but I cannot really make a conclusion based on that. So to measure genetic redundancy, I also looked at the replicate frequency spectrum. So what this is I would look at family 1. I identified the 100 SNP that have changed the most in this family. Then I look at how many animals have changed by, how many SNPs have changed by more than 0.3 in family 1. And then how many of those have changed in family 2, 3, 4, and 5. So a table will show me how they have changed. There are so many in this family and others, but it doesn't give me a nice indication of which ones changed similarly and which ones changed specifically in different families. So for that, I have this Venn diagram. So you can see of the 100 SNPs that changed the most in family 1, 15 of them are family specific, while 14 of them have changed similarly in all of the families. And there are 0 SNPs that have changed by more than 0.3 in any of the other families alone. When I look at the 100 SNPs in family 2, I see similar pattern where in family 2, there are ones that changed only in that family and none that have changed only in the others. The pattern persists with family 3 and family 5, but not with family 4. Here they have more overlapping SNPs, and there are other SNPs that have changed only in specific families. This we could also see from the previous inbreeding that cluster 4 was a bit more diverse and it had more animals. In conclusion, based on this study, is that I could find diversity within the breed, and I observed genetic redundancy within them. They have changed differently. This is good news for us because it allows continuous improvement in the population, which is a concern. It's been increasing a lot, and how much can we still continue? Further research is necessary to determine the application of what I found, because I looked at whether there are changes, but not much, how to deal with the changes or what it means for us. I'm hoping for more recent data. We are working on a contract to get the more recent data, and that would be much nicer because it would give me a greater number of animals for my older generations to base an allele frequency on. It could also give me a picture into more generations, which is nice because 10 generations is not a lot of time at all. This brings about other questions. So we treat the Holstein as a single breed, but is that appropriate? Should we treat it as different breeds? While I have shown that there's change, I have not determined exactly what caused what change. Could we find genes with this? I feel like we can, because if some markers change dramatically in all families, surely they're important to all. If they change in specific ones, they might be important for something specific. To truly see whether this happens, we would have to compare this to random situations, because some of these change will be due to drift, not necessarily selection. Are these families trait specific? I didn't have access to the milk records or genotypes for traits that we are really interested in, so I could not determine this. And how should we cluster? I used k-means clustering, but it's only one of the ways in which we can do this. Overall conclusions from my study was that there's not much benefit in multi-breed, but there's also not much loss with multi-breed. It's important that all breeds must be included in the reference population. And this is also reflected in within breed, that all should be included in the within breed reference population. Crossbreeds can be included in valuations. Some have done it in different ways. And there is underlying genetic diversity within a breed. And with that, I thank you for listening to my talk.
Yvette, thank you for a very nice talk on quite interesting topics. I assume we will have some questions. I'm going to open chat, and I think anybody can just unmute themselves to start asking questions. And maybe just a short one from me, based on your last research topic. So would you in future propose, I mean, if there's some additional variability within the breed that we don't know about, should we kind of exchange in future inbreeding rate with some other measure?
That is a good potential, and I've seen discussions in a recent email topic where people were wondering how exactly we should be doing this. And it is worth looking at. So inbreeding is one of those. And as you say, there's different ways and different tools have become available to us to do this. It's definitely worth looking at and comparing.
Okay, thank you. Well, there was quite a lot of people in today's talk, so I assumed we're going to have a bunch of questions. But
can I ask a question, Ida?
Mm-hmm.
Yeah,
yes,
please go ahead, Mike.
Okay, thanks. It's Mike Coffey here from SIUC. I only just caught the back end of your talk, I'm afraid, but your conclusions were interesting. There's not much benefit for including crossbreeds, but there's no disbenefit, so you may as well throw them in. And this is obviously in dairy cattle, but I'm just kind of wondering whether the same principle would hold if you were doing something like crossbred beef dairy cattle, you know, and something like feed intake in dairy cross beef cattle and feed intake in dairy cattle themselves. And if they were related, would there be any harm or benefit in incorporating beef with dairy?
I'm glad you asked that, so it gives me an opportunity to clarify. So those conclusions I made in the end is that there's not much benefit in multi-breeds over doing them separately, and there's not much loss. But when it comes to crossbreeds, there's been various studies that showed that it is important, especially something like beef on dairy, where focus is on crossbred performance. It is important to add those crossbreed animals. In other studies, Australian ones where they used crossbred sheep, it helped. If you included the crossbreds, it could increase the accuracy of the component purebreeds, and it helped for a crossbreed prediction of a breed that was not included in the reference population at all. In a study by Ivan, it didn't make a difference when he added the crossbred animals, but including with the eigenvalue, the numbers of eigenvalues. But in his case, most of the parents were genotyped and measured. So in the crossbreeds, it didn't make much of a difference, but that is not the case for everyone. So for me, it could help adding crossbreeds. And if your focus again is on crossbred performance, like the pigs, they have shown that it is important. You must include your crossbred animals. Mm-hmm. So it is important. The big question is for your mating strategy in your industry, how you should include them is the bigger question.
Okay, thanks very much, Yvette. Very interesting. Thank you.
Hi, I've got a question, if you don't mind. Hi. So really cool stuff. I'm especially intrigued by the last part, like with the detecting genetic redundancy. And I guess what I'm wondering is sort of because you started out introducing it by sort of describing genetic redundancy as I guess as a consequence of a standard quantitative genetic model. Right? You have a lot of loci, some loci, you know, some alleles go up in one family, other alleles might go up in another family. And I'm wondering, do you have any feel for if the patterns you see in the data, are they what you would expect simply from like an, you know, additive, massively polygenic trait? Or is there something sort of new and potentially more complicated going on with, you know, G by E or epistasis or whatever, right?
So for me, if I really wanted to look at that, I should compare it to one, dividing my selected candidates randomly into clusters, or just taking those initial genotypes and putting it in a simulation and do no selection at all. But looking at other studies, the pattern is quite similar, where there are small shifts for many of them. A complication for my study is within 10 generations, you don't see much. So many will show within 10 generations, it will shift a bit more and then it starts to reach an equilibrium and then it'll go to where it is more ideal. So outside that, you would see more. And something like epistasis that's a tricky one on exactly how to determine what, and there's epistatic effects everywhere and some are smaller. And I am looking forward to the World Conference and another conference I'm planning to go to recently, because I can be exposed to people who are more population geneticists and discuss this in better detail and learn from them, because I do see more potential. But as you say, some of these changes are similar to what I've seen from other simulation studies.
Cool. Thank you.
I hope I answered your question.
Yes, you did. And I'm also looking forward to the World Congress.
Yvette, we have a question in chat from Janis. Janis is working with dairy cattle, so probably interesting talk for him. He's asking, you shown that the approach where you have multiplied the breed proportion with the breed specific SNP effect was the least accurate. Do you have any ideas why this method was the least accurate in your study?
So for one unfortunate reason is that I realized this later, that I also should have weighted the intercept for the breeds, which I did not do, and that could have affected my accuracy. Unfortunately, once I realized this, I couldn't have access to the data anymore to go test it again. But I've discussed that with Paul van Reyden, and Ole Christensen also contacted me for that one. Another one I think is, as Paul van Reyden's study showed, is when the predominant breed was higher, you got higher accuracies, while in my situations, it was not so high. So among my accuracies, I would expect that proportion to be less accurate anyway. I think for those animals, the approach is simply too crude, because 50% is what 50% really. If it's 75%, it's a better estimate of there's at least one that should be more dominant. In Ole Christensen's study, it was Eriksson, so under lead of Ole. They used the breed proportions as well, and it was a bit more accurate, but taking breed origin of alleles into account was even better. So I definitely see the breed origin of alleles as a better way to approach this, especially if you can do it well. So there are caveats in different methods of how to do it.
Janis thanks you, as you can see from the chat.
Thanks.
Okay, we still have about 10 more minutes, so why not use the time? So go ahead with any other questions you have.
Yvette, hi, Gregor here. Can you hear me?
Yes, I can.
Yes, thank you for the talk. So did I hear just correctly that you mentioned that Ole and his colleagues, they found that breed of origin worked better than the breed proportion?
It did, yes.
Why is that? What's the rationale for this? Because in this area we see lots of observations, but I'm kind of lacking the theory. You know, I tried this, you tried that, someone else tried something else, but I'm kind of lacking some coherent theory about what should work and what shouldn't work, etc. So what's your hunch why, you know, the origin of alleles?
So for me, it's really different SNP effects can come from different populations. There's already a complication in adding heterosis and having different sections, but it'll also be bigger sections. So breed origin of allele could be over a bigger window that could have a similar effect coming from that breed and that breed. And I think it's overall simply just being less crude than breed proportions would already help, because it's not realistic to look at it so simply. So he used simulations using a dairy breed rotational cross. So in that case, there would also be a pure breed that's a bit more dominant than the other one with every haplotype. And pigs, they've done it as well. I think for pigs, it might even be more important. I'm not sure. I would have to read his paper again in very big detail to know that. And if he is in this group, it would be nice if he can share some information with that as well.
Yeah, thank you. I don't think he's here, but yeah. Thanks for the answer.
Okay. Do you have a theory?
No, I don't. That's why I'm wondering, you know, what we should be working on and developing some framework, and then obviously, you know, be testing different things within that framework.
And with some, like in other studies that I saw, using breed origin of alleles was not always worth the effort. The accuracies were only a little bit more. but the differences occurred more when the breeds were more different. If you're going to add dairy breeds that are more or less similar, there's not really a point in adding that. But I think adding something like a Holstein and a very small one, it could make a bigger difference. So sometimes it's worth analyzing whether it's worth the effort.
As always, sometimes theory says something, but then in practice you realize it's not really worth it. Yeah.
Yes.
Thanks.
So in that sense, what would be the future for the very small breeds?
The very small breeds. How small is very small, Ivan?
Yeah, that's another good question. Yeah.
Because I think for them, it becomes harmful if you start relying on genomic predictions when your collection already is not good. If you're not big enough to get good with in-breed evaluations, you should not be too optimistic. You should actually be a little bit afraid of genomic evaluations. Then even if you get included with a bigger breed, you still won't get much more accuracy than you would have with in-breed. So my big suggestion for them is first become big enough. And their advantage, though, if they start with genetic collection, is they would end up having genomic information about a lot of their breed, especially looking 10 years ahead, 20 years ahead. Then they will have a different kind of advantage, which would be very useful for studies, not just for genomic predictions.
Yvette, can I just jump in there and say then, I wonder if, let's say you had a small population of, say, Guernseys, and they, there was only a few thousand of them, and they genotyped most of their animals, but they're not making much progress with genomic evaluation. What if they had the capacity to create some Holstein cross Guernseys and genotyped those crosses, and then included their Guernsey and their Guernsey cross Holsteins in the Holstein genomic evaluation?
I believe that would work. I'm also thinking of, for example, the sheep breeds that came out, where it did help to bring those crossbreeds in. Because as you say, even though it's crossbreeds, it's still some Guernsey haplotypes that are coming into the system. It is more information on the Guernsey. Although the increase would not be as much as adding more Guernsey, it will definitely occur. And it might even, I don't have much to base this on, be better than adding Guernseys from a completely different country, especially because Guernseys are so small, they might differ more in different countries. Like I said, I didn't test that. I'm just thinking that might be.
Sure, that's in a funny kind of way. That's why I posed the question, because certainly in the UK, all of the available Guernseys have now been genotyped. So the only way they can increase effectively their reference population would be to strategically cross some Guernseys with Holsteins or with Jerseys and try to suck some information from some of the other breeds into their own evaluation through those crossbreds.
And you see good results?
I don't know yet is the answer. Myself and Gregor are working on this as we speak.
Great. So there's another topic to discuss at the World Conference when I meet you guys.
Yeah, I'll see you there.
Thank you. The UK especially has a lot of knowledge and work on crossbreeds. If you look at Ireland, which is not part of the UK, but they have so many genotypes and they combine them all. It's in their beef about 41 different breeds, although those breeds are at least also combinations of crosses, which makes that more reasonable. And I know the Irish people more than people from the UK, so I'm not sure how you are doing, but is your government also subsidizing genotyping like the Irish?
No, not at all. I mean, there are populations that manage to get research projects and get funding within the research projects to genotype their populations. But certainly in the bigger breeds, like the Holsteins and so on, and the bigger beef breeds and sheep breeds, it's all private money, as much as like it is in North America. The same. The Irish situation is unique within Europe, shall we say?
Thank you for a very nice discussion on the topic. So I would, okay, we have another question from Yanis, and I think then slowly we will go towards the end. So what would you suggest to a crossbreed population, genotype and phenotype animals to create its own reference population, or collaborate with purebred, use their reference and save some money? If you have chat, you can open to see the question.
I see, yes. I think
Perfect.
this question is also dependent on what exactly you want to do with this. Because if you look at crossbreed populations, to use them by themselves, that is good for an admixed population or more breed, like a complementary breed, where you want that group to actually be a group by itself. And I'm thinking of the Kiwi crosses, although I'm not sure whether that's exactly what they want to do. And in South Africa with composite breeds, where they want to be their own, that would be a case where you really want enough of your own to make it better. If there's good relationships with those peer breeds as well, that would be good. I would also advise that. Include the breeds that are part of your composite. If your strategy is like pigs, where you select your peer breeds to predict your crossbreeds, then you need all of those peer breeds. And there was a study that Yvonne Vinkis also did, or at least her group, where it was a strategy of selecting your reference population to also save money, whether you should have more of the crossbreds or more of the purebreds. And that one was quite interesting as well. That showed a bit more financial numbers. But again, as I say, this kind of depends on what exactly it is you want to achieve with your selection strategy. And whether you can collaborate with those people, because some associations are quite difficult.
Thank you. And we have a few more minutes, just so any final questions, please go ahead now. If not, I'm going to stop with recording right now.